Gregor John Mendel, who is called the father of genetics, started his famous hybridization experiments on garden pea, botanically known as Pisum sativum, in 1857 and published his results in 1866 in the form of laws of inheritance, also called laws of heredity. His laws of inheritance were in fact rediscovered in 1901 by Hugo de Vries from Holland, Karl Corrinus from Germany, and Erich von Tischermark from Austria. Mendel gave the concept of factor, which is now called gene. According to this concept, each character is controlled by a factor or a gene. For each character, there is always a pair of factors involved, one each contributed by male and female parent during reproduction. In the modern concept, genes are the structural and functional units lying on the chromosomes. Like chromosomes, they exist in pairs, one on each member of the chromosome, and determine the biological character of an individual. The two sister genes on a particular locus may or may not be identical and are technically known as alleles. Chemically, the genes are composed of deoxyribonucleic acid or DNA and direct the cell to perform a particular function through the process of transcription and translation. Mendel, in his experiments on hybridization, used 7 pairs of contrasting characters in pea plant, for example, plant height, pod length, pod position, flower color, seed color, seed shape, etc. Each of these 7 traits in pea plant is controlled by a gene with 2 alternative forms called alleles, one allele acting as dominant and the other one as recessive. The dominant allele can express in homozygous as well as in heterozygous condition, whereas the recessive allele can express only in homozygous condition. Based on his monohybrid and dihybrid crosses, Mendel proposed 3 laws of inheritance. Number 1, principle of segregation, which is also called law of purity of gametes. This law states that for each character there are 2 alleles, and at the time of gamete formation each allele enters into a separate gamete. Each gamete is thus pure for a particular type of allele. A random mating between these gametes restores the paired form of alleles. Number 2nd law is principle of dominance. For each pair of contrasting alleles, one allele acts as dominant and the other allele acts as recessive. The dominant allele expresses in F1 generation in homozygous as well as in heterozygous condition, whereas the recessive allele expresses in F2 generation only in homozygous recessive condition. Third law is principle of independent assortment. According to this law, the alleles of each trait segregate independently of each other and give rise to different gametes. Random mating between these gametes produces parental and recombinant progeny in F2 generation. In order to understand the law of independent assortment, let us discuss Mendel's original dihybrid cross in pea plant having 2 pairs of contrasting traits with seed shape and seed color. The 2 parents selected by Mendel with respect to seed shape and seed color were parent 1 having round and yellow seeds and parent 2 having wrinkled and green seeds. The plants with round and yellow seeds were crossed with plants having wrinkled and green seeds. All the F1 seeds obtained were round and yellow. When F1 plants were selfed, they produced a total of 556 F2 seeds, which were of 4 types: round and yellow 315 seeds, wrinkled and yellow 101 seeds, round and green 108 seeds, wrinkled and green 32 seeds. These 4 types of results were obtained by Mendel in the ratio of 9:3:3:1 respectively. In the F2 result, round and yellow seeds and wrinkled and green seeds are the parental characters, whereas round and green seeds and wrinkled and yellow seeds are the recombinants. Based on these observations, Mendel proposed the principle of independent assortment. According to this principle, the alleles of two pairs of contrasting traits with seed shape and seed color during gamete formation assort independently of each other and give rise to different combination of gametes, parental as well as recombinant types. Random mating between these gametes will produce different phenotypes in the ratio of 9:3:3:1 in a dihybrid cross. Now, let me explain this dihybrid cross in another way. Seed shape in pea plant is controlled by one gene with two alleles, capital R for round shape which is dominant and small r for wrinkled shape which is recessive. Similarly, seed color is controlled by another gene with two alleles, capital Y for yellow color which is dominant and small y for green color which is recessive. A dihybrid cross involving one parent with round and yellow seeds, that is capital R, capital Y, and other parent with wrinkled and green seeds, that is small r, small y, known as a dihybrid cross. Each parent in this dihybrid cross produces single type of gamete, that is capital R, capital Y type by one parent and small r, small y type by another parent. Fertilization between these gametes results into an F1 progeny in which all seeds are round and yellow. Because round is dominant over wrinkled, the F1 seed is however heterozygous in nature, that is genotype is capital R, capital Y, small y. When F1 plants are selfed, four types of gametes are produced. Number 1 capital R, capital Y, number 2 capital R, small y, number 3 small r, capital Y, number 4 small r, small y. The random mating between these gametes results in the formation of four phenotypes, viz. Round yellow, round green, wrinkled yellow and wrinkled green in the ratio of 9:3:3:1 in F2 generation. The occurrence of new combinations in this F2 generation, that is round green and wrinkled yellow, are only possible due to independent assortment of respective alleles followed by their random mating. The independent assortment of alleles occurs actually at anaphase stage of meiosis when the homologous chromosomes move to opposite poles. If the genes for seed shape and seed color are present on separate non-homologous chromosomes, the alleles of these two traits will move to anaphase in such a way that either two dominant alleles go to one pole and two recessive alleles go to the other pole. Alternatively, one dominant allele and one recessive allele may go to each pole. This is what is actually called independent assortment. Due to independent assortment, four types of gametes are produced: capital R, capital Y, capital R, small r, capital Y, and small r, small y types. Two of these types are parental, other two are recombinants. Random mating between these gametes will produce four phenotypes in F2 in the ratio of 9:3:3:1. Now, what is test cross? Test cross is a cross between F1 hybrid and homozygous recessive parent. A test cross here can be used to understand the mechanism of independent assortment. For example, when the F1 hybrid with round and yellow seeds having genotype capital R, capital Y, small y is crossed with a homozygous recessive parent small r, small y, that is wrinkled and green, the homozygous recessive parent here will produce only one type of gamete, that is small r, small y type, whereas the F1 hybrid capital R, capital Y, small y will produce four types of gametes. These four types of gametes are possible only due to independent assortment of alleles or genes. Then random mating between the male and female gametes will produce a phenotypic ratio of 1:1:1:1:1. The test cross result is thus here round yellow is to round green is to wrinkled yellow is to wrinkled green in the form of 1:1:1:1 ratio. W. S. Sutton and T. Bowery in 1903 showed a parallelism between the behavior of chromosomes and Mendelian factors. This can be explained, for example, number 1, the chromosomes occur in pairs like alleles of a gene. Number 2, the homologous chromosomes separate during meiosis like the alleles of a gene at the time of gamete formation. Number 3, different chromosomes segregate independently of each other during meiosis like different genes. Based on this parallelism, it was concluded that Mendelian factors were actually present on chromosomes, and this theory was called chromosome theory of inheritance by Sutton in 1903. According to this chromosome theory of inheritance, there is a strong resemblance between separation of homologous chromosomes during anaphase 1 of meiosis and the separation of alleles of different genes at the time of gamete formation. Each pair of homologous chromosome is oriented at metaphase plate independently of the other pair, and same is true for all pairs of homologous chromosomes in a complement. This orientation of bivalents at metaphase in turn determines the movement of chromosomes towards different poles at anaphase. Since genes are present on chromosomes, therefore the orientation of chromosomes at metaphase plate and their movement towards anaphase will determine the distribution of alleles into different gametes. Hence, there is a parallelism between the segregation of chromosomes and Mendel's principle of independent assortment. The independent assortment of genes for seed shape and seed color in pea plant in a dihybrid cross resulted into 9:3:3:1 dihybrid ratio and 1:1:1:1 test cross ratio. These two genes are now known to be present on chromosome 7 and 1 of pea plant respectively. These genes are in fact present on separate non-homologous chromosomes. The higher eukaryotes contain thousands of genes distributed on a set of few chromosomes. Therefore, it is certain that each chromosome carries hundreds of genes. So, two or more than two genes present on the same chromosome will tend to remain together during segregation and gamete formation. Such genes are then called linked genes, and the phenomenon of appearance of two genes together in the progeny is called linkage. The term linkage is genetically defined as the failure of two genes to assort independently. Linkage occurs when two genes are close to each other on the same chromosome. Chromosomes in turn are called linkage groups, as each chromosome contains a group of genes that are linked together. The number of linkage groups in any species is the number of types of chromosomes in that species. For example, a species having 2n is equal to 14 has 7 as the basic chromosome number. Therefore, in this species there are 7 linkage groups. The first evidence of linkage as an exception to independent assortment came from experiments with sweet pea, known as botanically named as Lathyrus odoratus. Bateson and Punnett, 1905, crossed two varieties of sweet pea that differed in flower color and pollen length. A sweet pea variety with purple flowers and long pollen, designated genotypically as P, capital L, was crossed with another variety with red flowers and round pollen, designated as small p, small p, small l, small l. All the F1 plants had purple flowers and long pollen. When F1 plants were self-fertilized, the F2 progeny produced purple flowers and long pollen with 583 individuals, purple flowers and short pollen with 26 individuals, red flowers and long pollen with 24 individuals, and red flowers and short pollen with 170 individuals. These individuals were obtained in an F2 ratio of 23:1:1:7. Thus, a phenotypic ratio of 23:1:1:7 was obtained by Bateson and Punnett instead of Mendelian 9:3:3:1 ratio. So, it is clearly a deviation from Mendelian dihybrid ratio. Similarly, when F1 plants with purple flowers and long pollen designated by capital P, small p, capital L, small l were crossed with a homozygous recessive parent with red flowers and round pollen, that is a test cross was performed. The test cross ratio obtained was 7:1:1:7 instead of 1:1:1:1 ratio as expected in case of Mendelian test cross. These dihybrid and test cross results clearly indicated that there was a tendency in the parental traits to inherit together in the progeny. In other words, the dominant or recessive alleles of these traits inherited together in the progeny. This tendency of dominant alleles to remain together in the progeny was called gametic coupling by Bateson and Punnett. In another situation, it was observed that when two such dominant alleles, that is capital P and capital L, come from different parents, they tend to remain separate. This was called as gametic repulsion. Thus, in Bateson-Punnett's experiment, the two dominant alleles in sweet pea and their recessive counterparts could be in coupling or repulsion phase. Bateson and Punnett, however, could not explain the exact reasons of coupling and repulsion. T. H. Morgan in 1910 studied the inheritance of three genes present on X chromosome of Drosophila, namely body color, eye color, and wing length. The dominant and recessive forms of these traits are, for example, in body color, gray body is dominant over yellow body. In case of eye color, red eye is dominant over white eye. In case of wing length, normal wings are dominant over miniature wings. It may be noted here that females have two X chromosomes, that is, they have XX condition, while male Drosophila have only one X chromosome, that is XY condition. Morgan in his experiment crossed a female parent with yellow body, white eye, and miniature wings, that is all alleles in recessive condition, with a wild type male with gray body, red eye, and normal wings, that is all genes in dominant condition. In F1 generation, all males were yellow body, white eyed, miniature wings, and all females were with wild type with gray body, red eyes, and normal wings. An intercross between F1 males and females gave the F2 progeny in which majority of the males and females were parental types. However, a significant number of recombinants were also observed by Morgan. Based on these observations, Morgan made three important conclusions. Number one, the genes for body color, eye color, and wing length are located on X chromosome of Drosophila and tend to be inherited together, that is, they are linked. Number second, due to crossing over, the homologous X chromosomes in females gave rise to the formation of recombinants. Number third, the likelihood of crossing over depends upon the distance between the two genes. That is, crossing over is more likely to occur between two genes that are far apart from each other. Morgan proposed that the genes are found in coupling or repulsion phase because they are present on the same chromosome in case of coupling, or on two different but homologous chromosomes in case of repulsion. Such genes are then called as linked, and the phenomenon is called linkage. According to Morgan, the two genes can be either close to each other or away from each other on a chromosome. Thus, the strength of linkage will be determined by the distance between the two genes in question. The greater this distance, lower will be the strength of linkage, and smaller this distance, the greater is the strength of linkage. The linkage is broken down by a phenomenon called as crossing over. Crossing over means exchange of chromosomal segments and occurs in prophase of meiosis I, when the homologous chromosomes are in paired condition. Crossing over results into reshuffling of genes, leading to the formation of recombinants. In case of weak linkage, crossing over occurs frequently, whereas it is rare in case of strong linkage. In order to understand the phenomenon of linkage and crossing over and differentiate between linkage and independent assortment, let us take a hypothetical example where two genes, capital A and B, are involved with small a and small b as their recessive alleles respectively. As you know, in case of independent assortment, the two genes are present on two non-homologous chromosomes. Independent assortment of these chromosomes will lead to the formation of four types of gametes in equal proportion. The expected frequency of a test cross progeny, therefore, will be in the ratio of 1:1:1:1. It may be noted that in such a test cross, 50% parental types and 50% recombinant types are produced. Now, in another situation, suppose that the genes A and B are present on the same chromosome. During a test cross, the homozygous recessive parent will produce only one type of gamete, that is small ab type, whereas in parent 1, the formation of gametes will depend upon whether crossing over takes place or not. If crossing over takes place, recombinant gametes are produced, but in case of no crossing over, parental types are produced. Since all types of gametes are possible in a population, therefore, test cross progeny will result into the formation of parental types as well as recombinants. The frequency of parental types will be, however, more, that is greater than 50%, as compared to recombinants, that is less than 50%. Such a test cross will therefore never produce 1:1:1:1 ratio as observed in case of independent assortment. Thus, genetic markers or genes located on the same chromosome tend to remain together during sexual reproduction and do not exhibit independent assortment. The genes are said to be linked whenever over 50% of gametes produced contain parental combinations of genes and less than 50% of gametes contain recombinant combinations of genes. The easiest way of finding out the proportion of parental and recombinant types is to make a test cross. The frequency of recombinants can be used to measure the intensity of linkage between the genes. Tightly linked genes seldom recombine, that is, the crossing over hardly occurs between such genes, and hence a low recombination frequency is obtained in a test cross. On the other hand, genes that are loosely linked, that is which are far away on a particular chromosome, they recombine frequently, that is crossing over occurs frequently between them, and hence a greater frequency of recombinants is obtained due to crossing over. But this frequency never exceeds 50%. Recombination frequency means total number of recombinants out of the total progeny produced. Recombination frequency helps us to understand how far two genes are close to each other on a chromosome. It determines the strength of linkage. More the recombination frequency, greater the distance between the two genes, because more are the chances of crossing over. Similarly, less the recombination frequency, more strongly the two genes are linked, because the chances of crossing over are less. As a rule, for any two genes, the frequency of recombinant gametes never exceeds 50%. This upper limit is reached when genes are very far on the chromosome. It is also reached when genes are on different chromosome and show independent assortment. Laws of inheritance were proposed by Gregor John Mendel. According to the principle of independent assortment, the alleles for contrasting traits segregate independent of each other during gamete formation. When the alleles or genes of two contrasting traits show independent assortment, a dihybrid ratio of 9:3:3:1 and a test cross ratio of 1:1:1:1 is expected. Independent assortment of genes is actually based on the movement of chromosomes towards different poles at anaphase and their subsequent incorporation into different gametes. Both parental and recombinant types are produced in the progeny. The concept of linkage was proposed by Morgan 1910 during his experiments in Drosophila. The genes present on the same chromosome tend to inherit together and are said to be linked. When two genes are linked, a completely different dihybrid and test cross ratios are obtained in the progeny. The strength of linkage is based on the distance between the genes. Linkage is broken by the phenomenon of crossing over. Thus, linked genes show independent assortment only after crossing over has occurred between them. Crossing over reshuffles the genes between two homologous chromosomes. The genetic consequences of crossing over are the formation of recombinants. Recombination frequency never exceeds 50% and is mostly less than 50%.